Hey everybody, welcome to week 6 of our class. This week we'll be covering chapter 9, which continues talking about genetics. Chapter 9 covers meiosis, and meiosis are how sperm and eggs are created. So meiosis serves two major functions. This includes reducing the number of chromosomes in each cell, and then also shuffling around the chromosomes in the cell to produce genetically different gametes, which again are eggs and sperm. Just a little reminder, homologous chromosomes, which are also called homologs, are going to be genetically similar in that they are both the same size and they carry the same genes. However, they may contain different alleles. So let's just do a brief overview of meiosis. And to start that, we will talk a little bit about the type of chromosomes found in each cell. So a child receives one homolog or chromosomes from each parent. And again, just as a reminder, homologous chromosomes are going to have the same genes, but may contain different alleles, which again is just an alternative form of alleles. Both males and females have 23 pairs of chromosomes, or a total of 46 chromosomes, which means that are also called diploid cells. And diploid cells is going to be represented by a 2 and a lowercase n. So pretty much all the cells in your body are going to be diploid cells. However, the gametes that are produced by both male and females are instead going to be haploid, which means that they are going to have half the amount of chromosomes as our normal body cells, also called autosomes. Haploid cells are going to be represented by a lowercase n. So just as a reminder, female sex chromosomes, or females and their sex chromosomes, are going to be X and X, and male chromosomes are going to be X and Y. The X chromosome is a lot bigger than the Y chromosome, and as a result, it's going to contain a lot more genes than the Y chromosomes. In mammals, the male will determine the sex of the offspring. However, in birds and some fish, the female will determine the offspring. These animals have different sex chromosomes, which are Z and W. The males are going to be WW, while the females are going to be ZW. However, the female chromosome is still going to be a lot bigger in these male, it's going to be a lot bigger than that male chromosome. This is the karyotype of a male. You can see the 23 pairs of homologous chromosomes, 22 pair of autosomes, and then the 1 pair of the sex chromosomes, which you can see in the bottom right-hand corner. They are labeled X and Y. Except the sex chromosomes, every one of the homologous chromosomes are going to be the same length, shape, and then the centromere is going to be in the same place. You get one homolog from your mom and then one homolog from your father. So mitosis and meiosis will play an important role in the life cycle of any animal. In the life cycle, in a sexually reproducing organism refers to all the reproductive events that occurs from one generation to the next. Mitosis is involved in the continuing growth of a child and the repair of tissue throughout life. For example, like if you cut yourself, your skin cells will undergo mitosis to cover up that cut so that so that no bacteria can get into your bloodstream and you could potentially get sick. As a result, all cells in the body are diploid, again meaning that they contain the 46 chromosomes. Meiosis, though, reduces the chromosome number by half, turning one diploid cell into 2 haploid cells. Gametes, which include egg and sperm, have only one member of each of the homologous pairs. Spermatogenesis means the production of sperm in males. Oogenesis is when the eggs are produced in the ovaries. And then, of course, upon fertilization, one sperm, haploid cell, will fertilize an egg, another haploid cell, to make one diploid cell. So here's the life cycle of a human, of course, which starts out with the fertilization of the egg by sperm to form a zygote that is diploid. The zygote then undergoes rapid cellular division using mitosis, which produces identical daughter cells to form a mature baby. During the human's life, the cells will use mitosis to allow the human to grow up into an adult and to repair damaged tissue. After puberty, the body uses meiosis to produce gametes, and then eggs and sperm are not identical to the parent. Instead, it will only contain half the chromosome of the parent cell, which is going to be 23 chromosomes. Now the egg and sperm are called haploid cells, and again, when the egg and the sperm come together, we now have a diploid cell. So like in mitosis, DNA is replicated at the beginning of meiosis. When the chromosome is replicated, it forms two sister chromatids that connect at the centromere. The centromere, again, is the protein that holds the sister chromatids together, and each sister chromatid will have identical DNA. So after the DNA has replicated, the cell will now undergo meiosis 1 and meiosis 2. So during meiosis 1, the homologous chromosomes will line up in the center of the cell and then be pulled apart to the poles of the cell, and then the cell will be separated. It will then quickly go into meiosis 2, and there is going to be no interphase between meiosis 1 and meiosis 2. And so there's going to be no DNA replication. The same thing is pretty much going to happen. The chromosomes are going to line up in the center of the cell, and now the sister chromatids are going to be separated. And then finally, the two daughter nuclei will separate. So we're going to end up with 4 daughter cells. So the beginning of meiosis is going to be very similar to that of mitosis. We're going to have the DNA replicating, the nuclear membrane is going to start dissolving, and then our spindles here are going to be forming. During meiosis 1, the homologous chromosomes will be placed right next to each other. So here we have one homologous chromosome, and then the other blue homologous chromosome. And then the spindles will attach to the centromere of the homologous chromosomes and then pull them to the poles of each cell. It will then undergo cytokinesis, and now we have 2 daughter cells. And then each daughter cell will undergo meiosis 2. Again, remember that there is going to be no interface between meiosis 1 and meiosis 2. So both daughter cells are now going to undergo meiosis 2. So again, kind of same thing. The chromosomes are going to line up in the center of the cell, although notice this time that they are stacked on top of each other rather than right next to each other as in meiosis 1. So our spindle fibers are going to connect to the centromere again. And now, instead of the homologous chromosomes being separated, now the sister chromatids will be separated. 2 daughter cells will undergo cytokinesis again, and now we have 4 cells that are not genetically similar, and the cells are now haploid. So both meiosis 1 and meiosis 2 are going to contain the same basic steps as mitosis. We're going to have prophase 1 and 2, metaphase 1 and 2, anaphase 1 and 2, and then telophase 1 and 2. So during prophase 1, the nuclear envelope dissolves and the replicated DNA condenses into chromosomes. The spindles are going to be forming, and then the homologous chromosomes will line up next to each other to form the tetrads and perform crossing over. Crossing over is the exchange of the genetic material between the homologous chromosomes, so that each sister chromatid is going to be different from one another. During metaphase 1, the homologous chromosomes line up right next to each other in the center of the cell. During anaphase 1, the homologous chromosomes are pulled apart to opposite sides of the cell. During telophase 1, the spindles detach from the centromere and the nuclear envelope forms, and then cytokinesis allows us to have two daughter cells. So after meiosis 1, meiosis 2 quickly starts. The events of meiosis 2 is going to be the exact same as that of mitosis, as you'll see here in a little bit. So during prophase 1, our spindles are forming, our nuclear envelope is dissolving, and we still have our chromosomes there that contain the two sister chromatids. Remember that between meiosis 1 and meiosis 2, the DNA has not replicated. So then we go right into metaphase, and this is where the chromosomes will stack on top of each other, instead of lining up right next to each other as in metaphase 1. The spindles now are going to connect to the centromere. And then during anaphase 2, the sister chromatids are now being pulled apart, as you can see here, to the opposite sides of the cell. And then telophase 2 is when those sister chromatids have reached the poles of the cell, and cytokinesis takes place quickly after, with the start of that cleavage furrow, and the cytokinesis will finish when we have our 4 daughter cells. So a couple slides ago, I mentioned crossing over, and this happens during prophase 1. And crossing over is really important in the formation of the gametes. So during crossing over, the homologous chromosomes will form something called a tetrad. It's called a tetrad, meaning 4, and 4 chromosomes are meaning that the 4 chromosomes are right next to each other. So the blue chromosomes and the red chromosomes here are homologous chromosomes. The blue chromosome contains 2 sister chromatids that have identical DNA, and this is going to be the same for the red chromosome. Notice here that the 2 homologous chromosomes are right next to each other, forming our tetrad. And the whole process of the homologous chromosomes forming a tetrad is called synapsis. So after a tetrad has formed, crossing over is going to occur, and this is when alleles from the same genes will be swapped between the homologous chromosomes. And this is very important, again, for making sure that the 4 sperm or eggs that meiosis is going to create is not genetically similar to one another. So here is our tetrad. Notice that the blue chromosome has 2 sister chromatids that are identical to each other, and then the red chromosome has 2 sister chromatids that are identical to each other. So during crossing over, this section of the blue chromosome will switch out alleles for this section of the red chromosome, and these 2 sections will contain the same genes. After crossing over has occurred, we now have 4 different chromatids, and these 4 different chromatids are going to end up in the 4 cells that we will end up with after meiosis is finished. So meiosis is going to be very important in the lives of any sexually reproducing animal. So by splitting the amount of chromosomes in our cells by half, it is ensured that when the sperm fertilizes the egg, the new organism will have the correct amount of chromosomes. If the baby has too many chromosomes or too few, it could result in various disorders or death even for the baby. Meiosis is also really important in ensuring that the genetic diversity of any species remains high. It achieves this through crossing over. Random fertilization also increases the genetic diversity of a species. Even without crossing over, random fertilization can produce over 7 trillion different human beings. So now let us compare meiosis and mitosis so that we can understand it a little bit more. So during meiosis, you're going to have two consecutive nuclear divisions, as we talked about a little earlier, meiosis 1 and meiosis 2. And then mitosis only has one nuclear division. Meiosis is going to produce 4 daughter nuclei, which ends up in 4 daughter cells after cytokinesis. And then mitosis will end up with 2 daughter nuclei, which is going to result in 2 daughter cells after cytokinesis. Following meiosis, the 4 daughter cells are now haploid, since it contains only half the amount of chromosomes as the parent cell. Mitosis, on the other hand, is going to contain the exact same amount of chromosomes as the parent cell, and is also going to be identical to the parent cell. Following meiosis, the 4 daughter cells will be genetically different from one another, but the daughter cells resulting from mitosis is going to be genetically identical to each other. So here we have meiosis represented on the left and mitosis represented on the right. Before each process, interphase will occur where the DNA will replicate. During prophase 1 of meiosis, the nuclear envelope will fragment and the chromosomes will connect. Synapsis and crossing over also occurs here. These, however, do not happen during prophase 1 in mitosis. During meiosis 1, the homologous chromosomes line up right next to each other. However, in metaphase 1 of mitosis, the homologous chromosomes will line up instead on top of each other. And this is going to be the same as in meiosis 2. During anaphase 1, the homologous chromosomes are going to be pulled apart from each other, as you can see here. However, in anaphase of mitosis, the sister chromatids instead will be pulled apart. The telophase is going to be the exact same for both processes. The chromosomes will reach the poles of the cell, the nuclear envelope develops, and cytokinesis quickly occurs. After meiosis 1, the cells are now haploid, meaning again that they contain half the amount of DNA as our original parent cells. Meiosis 2 and mitosis are going to be the same process, but instead meiosis ends up in 4 different cells, while mitosis ends up in 2 daughter cells. So both mitosis and meiosis is going to be really important in the lives of any sexually reproducing animal. Meiosis occurs during puberty of humans and only in the testes and the ovaries. Males can undergo meiosis all their lives, meaning that they can produce sperm all their lives. However, females cannot, as they only are able to produce eggs from the time that they reach puberty until the time that they reach menopause. Mitosis, though, is going to be much more common than meiosis. This will occur shortly after fertilization, so that the zygote will form into a fully developed baby, and then will occur as the baby develops into an adult. And then mitosis is also going to be really important in producing new healthy cells for each of the organs, and to help heal any wounds that the person may get. Just like with mitosis, it is very important that meiosis is done correctly during the formation of gametes. If the homologous chromosomes and sister chromatids do not correctly separate, then it will result in a disorder or even death for the human. And non-disjunction is when either the homologous chromosomes or the sister chromatids do not correctly separate. This results in either one or two of the daughter cells with an extra chromosome, and then one or two of the daughter cells with one less chromosome. The baby that forms from the gamete with one extra chromosome could develop a disorder called Down syndrome. And Down syndrome is called trisomy 21 because it will have 3 copies of chromosome 21. This will result in significant changes and could potentially involve mental disabilities. The chance of a woman having a Down syndrome baby grows significantly over the age of 40. And 80% of Down syndrome cases is a result of non-disjunction in the egg, and only 20% in sperm. So when non-disjunction happens during a meiosis 1, we're going to end up with both homologous chromosomes in one of the daughter cells. And this is because the homologous chromosomes were not separated correctly during the anaphase process of meiosis 1. However, during meiosis 2, the sister chromatids are able to correctly separate, and so we're going to end up with 2 gametes that will have 1 less chromosome, and we'll have 2 gametes that will end up with 1 extra chromosome. All of the gametes here will produce a baby with some type of disorder. So if non-disjunction happens during meiosis 2, then the sister chromatids of one of the chromosomes will not be correctly separated. This will result in 2 normal gametes, 1 gamete with 1 less chromosome, and then 1 gamete with 1 extra chromosome. 50% of these gametes will produce healthy babies, while the other 50% of the gametes will result in a baby with some type of disorder. So if non-disjunction happens with the sex chromosome, then it could result in either the person having 1 or 3 sex chromosomes. Newborns with an abnormal amount of sex chromosomes are going to be much more likely to survive than those with an abnormal amount of the other 22 chromosomes. This is because only 1 X chromosome is active in the cells of a person at any one time. Therefore, if the person receives an extra X chromosome, it will be inactivated in the cell, and then is called a bar body. However, if a baby only receives 1 Y chromosome, then it will not be able to survive. However, the baby will be able to survive if it gets only if it only receives 1 X chromosome. This is called Turner syndrome, and the baby will have female parts but won't develop secondary sex characteristics. If a person with 2 X chromosomes gets a Y chromosome, the dominant Y chromosome will overpower the X chromosome, so the baby will be a male, but with more feminine characteristics. This is called Klinefelter syndrome. The lady on the left-hand side has something called Turner syndrome, which is an example of monosomy since she will only have 1 X chromosome. This person will be female as she will still have ovaries. However, these ovaries will be underdeveloped and her breasts also will be underdeveloped. However, females with Turner syndrome could still get pregnant with in vitro fertilization. The guy on the left-hand side has Klinefelter syndrome, since he will have 1 extra sex chromosome. However, the sex chromosome will turn into a bar body. This person will still be male, but will have less developed testes, no facial hair, some breasts, and will have very long arms and legs. Alright guys, thank you for joining me. Let me know if you guys have any questions, and I hope you guys have a good week. See you later.